Hello, my name is Dr. Minde, and in this lecture series, we're going to discuss the microscopic organization of lymphoid tissue or lymphoid organs. So, the immune system is a system that plays a role of distinguishing self from non-self, or self from foreign substances. So, it's able to neutralize or inactivate foreign molecules, and this include microbes such as viruses, bacteria, parasites, and the immune tissue also destroys other cells such as virally infected cells or cancer cells. And in this case, you need to appreciate that we also have autoimmune diseases, and these are diseases that occur when the body's immune system starts attacking the normal body cells. And in that case, there's a problem. Immune cells are supposed to only attack non-self or foreign substances or cells that are infected or cancer cells. But when the immune cells now start targeting the normal body cells, those are what we refer to as autoimmune diseases. So the cells of the immune system are distributed throughout the body, scattered throughout the body, or they may be arranged in spherical nodules, and these form what you call lymphoid nodules. And the third way in which they are organized, lymphoid organs can form lymph nodes, spleen, thymus, or bone marrow. This is where they are organized into lymphoid organs. So they could be scattered throughout the body or form small spherical nodules or actually form lymphoid organs. So lymphoid nodules and isolated cells of the immune system form what you call mucosa-associated lymphoid tissue in the mucosa of different systems in the body, the GIT, the respiratory system, reproductive system, urinary system. So within the mucosa of these parts, viscera within these systems, you have lymphoid tissue, and we call them mucosa-associated lymphoid tissue. In the digestive system specifically, we have the tonsils, the pairs patches in the tonsils that are located in the oropharynx, the pairs patches in the ileum, and the appendix also has lymphoid tissue. So this is just the organization. This is where you have cervical lymph nodes here. You can appreciate the thymus is a lymphoid organ, the spleen is also a lymphoid organ, lymph nodes in the inguinal region, in the popliteal region, in the axilla, and so on and so forth. So you need to understand that we have an antigen and an antibody. An antigen is a molecule on the cell surface that is usually recognized by the immune system, while an antibody is actually a glycoprotein that is produced by immune cell that usually interacts with the antigen that is on the cell surface. So antibodies belong to the immunoglobulin family. So they are glycoproteins on the immune cells that are and interact with the antigens. So we have different cells in the immune system. We have the lymphocytes, plasma cells, mast cells, neutrophils, eosinophils, basophils, and the cells of the mononuclear phagocytic system. We also have antigen-presenting cells such as macrophages and dendritic cells. So we have three types of lymphocytes: B lymphocytes, T lymphocytes, and natural killer cells. B and T lymphocytes have the ability to selectively recognize a specific epitope. So they have the ability to recognize a specific epitope, and that is based on the receptor that is on their cell surface. So they differ based on the receptor on their cell surface. The B cell usually originates in the bone marrow and matures in the bone marrow. However, the T cell originates in the bone marrow, but it has to leave the bone marrow and go to the thymus, where it will proliferate and mature. Others, of course, will die by apoptosis. So both of them, B and T cells, produced in the bone marrow. Maturation of B cells is in the bone marrow, while that of T cells is in the thymus. So we have primary and central lymphoid organs. These are mainly the bone marrow and the thymus, where production and maturation of these cells are done. So those are the primary or the bone marrow and thymus form now the primary and central lymphoid organs. Then we go to the secondary or peripheral lymphoid organs. This is now where other immune cells and lymphoid tissue is found. So we have spleen, lymph nodes, the tonsils, appendix, and pairs patches of the ileum. So B and T cells usually are not uniformly distributed. They actually occupy preferential sites in these peripheral lymphoid organs. Natural killer cells are also types of lymphocytes. How are they different from B and T cells? They lack that receptor molecule that is a marker B and T cells have receptor molecules on their surface, and that gives them that is what recognizes them. But natural killer cells are lymphocytes that lack that marker, and 10 to 15% of lymphocytes circulating in the blood are actually natural killer cells. What is the function of natural killer cells? They are the ones that attack virally infected cells, cancer cells, or cells that have been transplanted. So they are involved in what we call the innate immune response, attacking virally infected cells, cancer cells, and transplanted cells. So this just shows you the bone marrow where T cells, B cells, natural killer cells, all of them are produced in the bone marrow. Then maturation of B cells and natural killer cells will still occur in the bone marrow, but the T cells have to go to the thymus to mature before they form the CD4, which are the T helper cells, and the CD8, which are the cytotoxic T cells. So we have different organs of the immune system. The thymus, which is located in the anterior mediastinum of the thorax, and it's a site for T cell maturation. Then we have the bone marrow, which is actually the home of the lymphocyte system, where the lymphocytes are produced and maturation of B cells occurs. And then we have the lymph nodes, which are found at major junctions of lymphatic vessels. So it is at the lymph nodes where both T and B cells are able to interact with antigen and antigen presenting cells. So if there's an infection, for example, on the upper limb, the microbes that will enter the body will be taken up by macrophages, which are antigen presenting cells, and these are going to present to T and B cells within the lymph nodes, for example, the axillary lymph nodes. So lymph nodes are a site where T cells and B cells interact with antigens and antigen presenting cells. Then we have the spleen. The spleen is located in the lower upper quadrant, in the left upper quadrant of the abdomen. And again, it's a place where T and B cells interact with antigens in blood. After that, we have the mucosa-associated lymphoid tissue. This is lymphoid tissue in the mucosa of different body systems. So like in the oropharynx, we have the tonsils, then adenoids in the nasopharynx, the Peyer's patches in the ileum, okay? And we also have mucosa-associated lymphoid tissue in the mucosa of the respiratory tract and the and the genitourinary tract. So these usually respond to antigens that enter the body through the mucosa of these systems. So if you're to ingest food that contains microbes, it is the lymphoid tissue that is within this mucosa-associated lymphoid tissue in the mucosa of the GI tract that will deal with that microbe first. So this just shows you again, this is the thymus in the anterior mediastinum, the spleen in the left upper quadrant. Okay, then you can see inguinal lymph nodes. You have some nodes in the cubital fossa, in the axilla, cervical lymph nodes. You can appreciate the adenoids in the nasopharynx and the tonsils in the oropharynx. So lymphoid tissue usually is just connective tissue that has rich supply of lymphocytes. So it's formed by connective tissue with rich supply of lymphocytes, and they can exist freely, as we had said, scattered cells or within the mucosa, or they can be surrounded by capsules to form lymphoid organs. Usually, lymphoid tissue are made up of free cells and a rich network of reticular fibrils. So collagen type 3 and free lymphocytes. So that's what form the lymphoid tissue. So the thymus is a lymphoepithelial organ. It's located in the anterior mediastinum of the thorax, and it is usually active during childhood. When you reach puberty, the thymus usually involutes, meaning it is replaced by adipose tissue. So the cells of the thymus include mainly the lymphocytes, T lymphocytes that arise in the bone marrow, and we also have epithelial cells, which are called the nurse cells. These nurse cells are the mechanical support for the lymphocyte population. The thymus towards the center has an epithelial framework of coarse, which is usually coarse and contains few lymphocytes. So towards the center, the lymphocytes are less, but towards the periphery, around the capsule, the lymphocytes are many. So from the capsule sends intrabacular to penetrate the parenchyma and divide the parenchyma into incomplete lobules. Then each of these incomplete lobule has an outer cortex, which is highly cellular, and an inner medulla, which is less cellular. So each lobule has a peripheral or outer cortex, which is highly cellular and dark. So the nucleus will take in the stain, and then a central or inner medulla, which is light staining and contains less number of cells. So the thymus has an outer capsule that sends in septa to divide the gland into lobules, so you can appreciate the lobules, and these septa are carrying with them neurovascular structures. Each lobule is made up of an outer or peripheral dark staining, highly cellular cortex, and an inner light staining central medulla. So within the cortex, we have the cortical epithelial cells, which are also called the NAS cells. And we said they provide mechanical support for the lymphocytes and also promote T cell differentiation and proliferation. So these NAS cells also secrete hormones that regulate T cell maturation and proliferation. So the thymus has two cells. We've said the T cells and the NAS cells. The NAS cells provide support. They promote T cell differentiation and proliferation, and usually they produce hormones that will regulate T cell maturation and proliferation. So the inner surface of the capsule, you remember I said the thymus has a capsule that sends in septa to divide the glands into lobules. So the inner surface of the capsule has these epithelial cells which lie on a basement membrane. Now we have what we call blood-thymus barrier. What forms blood-thymus barrier? These epithelial cells lying on the basement membrane, surrounding capillary endothelial cells lying on the basement membrane. Those form the blood-thymus barrier. So the two basement membrane of epithelial cells and the capillary endothelial cells, so plus the cells involved, they form blood-thymus barrier, and this barrier is able to protect the parenchyma of the thymus from antigenic material that are in blood. So blood has antigenic material or microbes. To prevent these microbes from entering the parenchyma of the thymus, you have blood-thymus barrier. This barrier is formed by three things: the fused basement membrane, as well as endothelial cells of the capillaries and the epithelial NAS cells of the thymus. So this is an infant thymus. Its capsule sends in septa to divide the gland into lobules. Each lobule has an outer dark staining, highly cellular cortex and an inner light staining, less cellular medulla. And these connective tissue septa have the neurovascular structures around them. Then this is a middle-aged adult thymus. You can appreciate the involution. The cells have been depleted and replaced by adipose tissue. Then this is the cortex. We said it is highly cellular, dark staining, contains immature cells, the thymocytes, and as they mature, they get towards the medulla. So the outer cortex has large lymphocytes that are dividing. So they have features of division, active mitosis. So as they mature up, they get towards the medulla, and the cells now acquire the surface markers to either become T helper cells or cytotoxic T cells. Then within the medulla, you have epithelial cells that form, they have large nuclei and eosinophilic cytoplasms with prominent basement membrane. And these epithelial cells, they become flattened and concentrically arranged with keratin filaments, and that's what forms Hustle corpuscles. So you can appreciate this is a Hustle corpuscles, where you have flattened epithelial reticular cells, usually arranged concentrically, and they're usually filled with keratin filaments. So the medulla is formed of cells that are light staining, mainly the epithelial cells, and they are flattened, filled with keratin to form the Hustle corpuscles. And usually the function of this is unknown, but they tend to calcify. What are the functions of the thymus? The main function is to help with maturation of the T lymphocytes. They also help to develop an immunological self-tolerance, where you're able to tolerate self, so that you don't, the body does not start acting on its own cells. Then the thymus also has the epithelial NAS cells that produce paracrine secretions. So the hormones and cytokines by epithelial NAS cells are paracrine secretions that will regulate T cell maturation and proliferation. These secretions include thymidine, thymopoietin, and thymosin, as well as thymus humoral factor. So these are the secretions to help with T cell maturation and proliferation. The thymus also produces proteins that act as growth factors. So the thymus receives its blood supply from internal thoracic and inferior thyroid vessels, as well as post-capillary venules that will allow the passage of lymphocytes into and out of the thymus. Remember, there are no afferent lymphatics on the thymus. Thank you very much.